Hello everyone, welcome to the St. Joseph's open web-based learning platform, that is how you learn. Also welcome to Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. Will be offered at the UG level.
Welcome back to the Plant and Animal Biotechnology course. That's the first module, that plant tissue culture and techniques and secondary metabolites production. The chapter 7 deals with the applications of plant tissue culture, synthetic seed production and cryopreservation. Wonderful applications of the plant tissue culture and the plant biotechnology especially. So these are the learning outcomes for the students. Will be able to be aware of the concept of the synthetic seed, what is synthetic seed, and they can easily assess the different methods for synthetic seed production, importance and scope of the synthetic seed, understand the role of various cryoprotectants in case of the cryopreservation process. So variety of cryoprotectants will be evaluated. Familiar, they are very familiar with the vitrification process, stabilization, and recognize the factors involved in plant regeneration and recovery. And these are the contents of the present topic, that is cryopreservation and synthetic seed and cryopreservation, concept of synthetic seed and its production, methods for producing synthetic seeds, scope and applications of synthetic seeds, concept of cryopreservation and cryoprotectants, factors influencing plant regeneration and recovery, applications and future prospects. So let us see first what is the synthetic seed, how it will be produced. It can be defined as artificial encapsulation of somatic embryo, shoot beds or aggregates of cell or any tissue which has the ability to form as a plant in vitro or ex vitro condition. So it's a wonderful application in the plant tissue culture. Toshio Murashige, first of all in 1970, coined the term synthetic seed. Look at the picture. This is the somatic embryo, which is surrounded with a seed coat, cotyledon embryos. Okay, yeah. So it includes the production. Maintaining genetic variation are difficult. Many fruit crops are difficult to produce seed, and difficult in dioecious plant for female plant. Seed-borne disease and pest. Some seedless fruits cannot be propagated through seed. Dormancy of the seed. So these are the limitations for preparing the synthetic seeds. Why natural seeds are important? Why can't they may not sustain in some cases? And this is the possible protocol for preparing the synthetic seed. The growth hormone, nutrient elements, explants will be included, gel essence, and additional materials such as antibiotics and antifungals also included in the bead structure where the seed can be protected. So these are the components used in synthetic seed production: gelling agents, complexion agent, coating agents, protective antibiotics, growth regulators. First of all, let us see what are the gelling agents. Sodium alginate, carrageenan, locus, bean gum, gelrite, agar, carboxy, methyl cellulose, sodium pactate, and tragacanth gum. So these are the gelling. Among these, sodium alginate is the best choice. After that, it follows the agar or CMC, also very important, and sodium pactate also gives the prominent results. Complex nascent. After preparing the gel suspension, calcium chloride, potassium chloride, ammonium chloride. So these are the mineral salts that can ensure the growth and healthy hygienity of the seed. Coating agents, after all, polylysine, LVAX 4260, gamma-3ZS, glutaraldehyde, malic anhydride, sodium alginate. Again, the sodium alginate with double concentration in the production of this synthetic seed. Rifampicin, cefotaxime, tetracycline HCl, mycorrhiza, activated charcoal are the antibiotics to protect the seed from the contamination chances. And the growth regulator including gibberellic acid, abscisic acid, zeatin, kinetin, benzylaminopurin, 2,4-dichlorophenoxyestic acid, indolactic acid, naphthalene acetic, and thiazide. So these are the growth regulators depending upon the species, depending upon the cell types, and seed coat, the combinations will be various. And this is the method for producing synthetic seed. First of all, you need to select the plant material and surface sterilized. Then somatic embryogenesis must be established. Okay, the plant, right? Then selection of mature embryo. Here the embryo and the stimulation of the variety of combination, that is high concentration of BAP, less concentration of the cytokinin can give rise to the torpedo shaped somatic embryo. The somatic embryos are isolated from embryogenesis mixed with a 0.5 to 5% sodium alginate solution, and then slowly 30 to 100 millimolar concentration of calcium nitrate solution is added to form the beaded like structures around the embryonated structures that forms the encapsulation, because the hydrophobic and hydrophilic environments will be there. That is encapsulation process. So once encapsulation process is done, each bead is carefully taken and tested on the embryo enhancement media concentrations, and then the seeds, once they develop the radicals, they will be carefully isolated or subcultured onto the vitrification process, and germinating and cotyledonous stage gives rise to plant completely developed into plant. And these may not be directly taken into the ordinary sunlight because they are very tender in condition, so that they must be taken and maintained under greenhouse chamber for evaluating the any pest associated or disease resistance or any abiotic and abiotic stresses are there in the plantlets. So why we need to synthetic seed production? So characteristics of clonal propagation system: micro propagation, low volume, small scale propagation method, maintains genetic uniformity of the plantations, accumulatize plantlets required prior to field planting, high cost per plantlet, relieve low multiplication rate, high volume, large scale propagation method, direct delivery of propagules to the field, thus eliminating transplants, low cost per plantlet, rapid multiplication of plantlet. So these are the reasons why the synthetic seeds are required. So this is the scope of the synthetic seed productions: propagation, conservation, transport. Rare and endangered plants can be propagated through the synthetic seed. Elite genotypes can be maintained under aseptic condition. Genetically engineered plants can be prepared. Seedless plants can be prepared. I mean, edible portions can be enhanced. I mean, juicy part or homologous juicy part, commercially important plantations can be enhanced to this synthetic seed production. Next, conservation. Short-term medium will be used, or long-term storage, or short-term medium. Slow growth conservation maintains under reduced temperature. Use of growth retardants such as abscisic acid, use of minimal growth medium, use of osmotic pressure, that is 10% of the mannitol solutions can be used. Reduction in the oxygen concentration, which will be the limiting factor for the synthetic seed germination process. The combination of more than one treatment that gives the more reliable synthetic seeds in commercial zones. And long-term cryopreservation is the best choice. Encapsulate, dehydrate, and vitrified synthetic seeds based on cryopreserved protectants, DMSO solution, and then freeze them. Subsequent freezing the synthetic seeds for longer period of preservation. Transport exchange of axinic. Axinic means new cultures. Plant materials free of undesirable contaminants will be possible through the synthetic seed production. So these are the applications of synthetic seeds. Hybrid plants can easily be propagated. Genetically modified plant or crops can be propagated. Endangered species can be propagated. Elite genotypes can be preserved or propagated. Synthetic seeds can be directly used in fields. Whether they are germinated hypocotyl radicals, if they are generated, they can easily be sown onto the crop fields, normal crop fields. Synthetic seeds can be transported from one country to the other without obligations, because short-term or long-term preservation methods are available. In this case, for country to country or region to region, cryopreservation is the best choice to send out the synthetic seeds. Cereals, fruits, medicinal plants can be studied anywhere in the world, because through the synthetic seed technology. Synthetic seeds are small, so they are easy to handle, easy to manage. Due to encapsulation, plant material gets the aseptic condition inside the capsule. During the transportation. During the transportation, synthetic seeds must not be agitated. That is the disadvantage with the synthetic seed. The next topic, that is the concept of cryopreservation, how cryoprotectants are useful. In Greek terminology, cryos means frosting, literally means in the frozen state. The principle involved the cryopreservation to bring the plant cells and tissue cultures to a zero metabolic rate or non-dividing state by reducing the temperature in the presence of cryopreservation. Over a solid carbon dioxide at 79 degrees centigrade, low temperature deep freezer at minus 80 degrees centigrade, and liquid nitrogen at minus 196 degrees centigrade. So example, plantations, rice, wheat, peanut and coconut germplasms and seeds can also be preserved through the cryopreservation. The mechanism involves the freeze preservation based on the transfer of water present in the cells from the liquid to solid state. First of all, sublimation condition. Due to the presence of salts and other organic molecules in the cells, the cell water requires much more lower temperature to freeze, even up to minus 68 degrees centigrade, compared to the freezing point of pure water around 0 degrees centigrade. When stored at low temperature, the metabolic process and biological deteriorations in the cells, tissues almost come to stand still. So that is the best option, best choice in case of cryopreservation. And need for the cryopreservation, development of sterile tissue culture. Addition of involves the mechanism involves the first of all, develop the sterile tissue culture. Addition of cryoprotectant or pre-treatment, freezing, storage, thawing, reculture. That is the simple way of the cryopreservation process. Measurement of survival or viability and plant regeneration. These are the steps involved in the mechanism of the cryopreservation. First one, the selection of plant species and the tissue with particular references to morphological and physiological characteristics largely influence the ability of the plants to survive in the cryopreservation. Any tissue from a plant can be used for cryopreservation. Example, merostems, embryos, endosperm, ovules, seeds, culture plants can be easily. Cryoprotectant addition. Cryoprotectants usually DMSO, glycerol, ethylene, propylene, sucrose, mannose, glucose can also be used as a cryoprotectant, but higher concentrations without contaminations. In freezing, it must be start from slow conditions, starting progressively reducing the temperature. Slow freezing method, rapid freezing method, stepwise dry freezing method are involving the freezing method. Maintaining frozen storage. Maintenance of frozen cultures at specific temperature is an important as freezing, very important aspect in the cryopreservation. Generally, the frozen cells or tissues are kept for storage at temperature in the range of minus 72 to minus 196 degrees centigrade. A protective clothes and protective gloves, goggles, and gloves must be worn during the cryopreservation process. Storage is ideally done in liquid nitrogen refrigerator at 150 degrees centigrade in the vapor phase or at minus 196 in the liquid phase, right? So, and thawing the final is usually carried out by plunging the frozen sample in ampoules into the warm water. So temperature 35 to 45 degrees centigrade bath with rigorous swelling. By this approach, rapid thawing at the rate of 500 to 750 degrees centigrade minute per liter occurs, and this protects the cells from the damaging effects ice crystal formation. So thawing is the process to reculture the whatever cryopreserved materials in the other region. As the thawing occurs, ice completely melts. The ampoules are quickly transferred to water bath at a temperature 20 to 25 degrees. This transfer is necessary since the cells get damaged if left for long warm bath condition. Reculture. In general, thawed germplasm is washed several times to remove the cryoprotectant. If any cryoprotectant, for example, increased concentration of dimethyl sulfoxide, increased concentration of sucrose maybe creates the negative pressure on the germplasm. The material is then cultured in a fresh media. Plantlet regeneration. The ultimate purpose of the cryopreservation of germplasm is to regenerate the desired plant for appropriate plant growth and regeneration. The cryopreserved cells or a tissue have to be carefully nursed and drawn. Addition of certain growth promoting substances besides maintenance of appropriate environmental conditions often necessary for successful plant regeneration. So these are the places, global seed vault near to Norway and Greenland, site of seed vault. All the viable seeds across the world are collected and preserved through the cryopreservation process for the future generations. These are the applications. Currently has a capacity to conserve 4.5 million seed samples with approximately 1.5 million distinct seed samples of agriculture crops thought to exert. The Salmod seed bank can store roughly 3 of each sample. In India, germplasm maintains and exchanges centers for agriculture and horticulture crops. NBPZR, National Board for Plant Germplasm Research, New Delhi, established in 1976. For medicinal and aromatics plants, botanical plants, BSA, Botanical Survey of India, Kolkata, West Bengal, established in 1890. The forest trees, germplasm, Forest Research Institute, that is Dehradun, Uttarakhand, was the optimal centers where the cryopreservations can be possible. So these are the applications. Gametes, embryos can be preserved without genetic drift, and it safeguards genetic integrity and valuable strengths, offers generation time, and allows further contribution of genetics. Eases the transportation of genetic stock since they are preserved ampoules and solidified states, and causes decrease of disease transmission since the low temperature won't allow the contamination of bacteria, fungal contamination. More or less after thawing or reculture, during the thawing culture, care must be taken if any personal unhygienic may create the problem to the ampoules. And these are the references for the cryopreservation and the present topic, synthetic seeds and the cryopreservation process. Thank you for your attention for the present topic, synthetic seed production and cryopreservation. Thank you very much.